Siobhan Ferguson, Bio 225, Week 2. This is aseptic technique and culturing microbes. Today is July 13, 2019. So for this experiment, I'm going to take the results from my microbes from sitting in my incubation area for 60 hours, and I'm going to transfer them into their agar plates. And I have my inoculation loops in alcohol. So first thing I'm going to do is light my fire so I can sterilize the tips of each tube. So I'm going to take the E. coli first and take the top off and sterilize it in the fire. And then I'm going to take my inoculation loop out the alcohol, shake it off so it's dry, dip it in here. And spread this into the first quadrant of the E. coli plate. I'm going to put that back in my alcohol. I'm going to sterilize the E. coli again and then put the top on it. And I'm going to take a second inoculation loop, shake it off, and spread the same microbe into the second quadrant of the APGAR plate. And replace that back into the alcohol. And take a third one, shake it off, and place it into the third quadrant. And then again, move this into the fourth quadrant. And cover it back up. And that is it for my E. coli. And I have labeled it so I know that it's E. coli. Now onto the S. epidermis. I'm going to remove the lid. Sterilize the lid. Take my loop. Spread this into the first quadrant of the corresponding APGAR plate. And we want to sterilize this again and put the top back on. And then we're going to take my second one and spread this into the second quadrant. And then my third. Spread this into my third quadrant. And then onto the fourth quadrant. And that is it for my S. epidermis. And I'm going to do the same thing for the S. cerevisiae. I'm going to uncap it. Sterilize the lip. And stick my inoculation loop in. Spread this into the first quadrant. I'm going to sterilize again and replace the lid. Take my second one. Spread this into quadrant 2. Third one. Spread into quadrant 4. I'm sorry, into quadrant 3. And lastly, take this and spread it to quadrant 4. And now we'll take these and place them back into my incubation area for 48 hours, and we'll come back and see the results. For my cleanup, I will dispose of everything down the sink, and of course, blow out my candle and clean my surface with 10% bleach. Bye. Siobhan Ferguson. Today is July 13, 2019. This is Bio 225, week 2, aseptic technique and culturing microbes. Results. So, my E. coli is cloudy, and there's a little bit of substance at the bottom, but it's mostly cloudy. My S. epidermis is clear, but there's also a little substance at the bottom of the tube. And the S. cerevisiae is just clear. And there's that at the bottom of the tube. Siobhan Ferguson, Bio 225. This is a week 2's lab. These are the results from my culture sitting in incubation for 40 hours. So for the E. coli, well, for all of them, they all look flat. The E. coli, the shape is irregular, the margins are entire, the elevation's flat, and this size is pinpoint. For the S. epidermis, it's rhizoid shaped. The margins are also entire. All of them have entire margins, and they're all flat. And the size is small. And for the S. cerevisiae, the shape is circular, margins entire, elevation's flat, and the size is puntiform. Puntiform or punctiform. These are the results.